So welcome everyone to a Genomics Aotearoa seminar. Nice to see you all here. Again, I say thanks to all you people who are in areas which are slightly shut down because of COVID. I hope things will be better soon. Fingers crossed. And today's speaker is one of the key people who has been making things reasonably not too bad. Today's speaker is Joep de Ligt. Joep did his PhD at oh god, I have to pronounce these places. Radboud University in the Netherlands, and then worked at the Hubrecht Institute, University Medical Centre in Utrecht, before coming to us in New Zealand in 2019, where he works for ESR in Wairarapa, which. Yeah. I remember a good friend of mine saying, how is it that we possibly managed to attract somebody of such skill to come to New Zealand? So I hope Joep is a key part of Genomics Aotearoa, leading many of our programs, but also has been a key part in the genome sequencing work around COVID-19, which I think has been one of the huge success stories of the response to COVID in New Zealand. And I hope he's going to touch on that and some other things. So I'm going to let you share your slides and get on with it. If you could you turn off your microphones and cameras so we have a bit of bandwidth. If you've got any questions, feel free to throw them in the chat during the talk. Otherwise, we can address them at the end. Otherwise, take it away.
Thank you, Peter. Tēnā koutou, e mihi ana ki ngā tohu nehe, o tawa e noho ana ni au. Ko Joep de Ligt tōku ingoa, ke te mahi o ki ESR, tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you very much, Peter, for that lovely intro. And yes, I have dabbled in many different genome sizes. And today, since there are a lot of people in GA that care a lot about big genomes, I thought I needed to make the point that tiny genomes are still worth something, especially when you can actually make a difference to people's lives with them. So I will be focusing on what we have been doing on the coronavirus. And if there's any questions, as Peter says, feel free to pop them in the chat and we will take them along a ride. There is one disclaimer that I have to give you, that is that today is a bit of a special day for book nerds around the world, at least if you're a fan of the Wheel of Time series, since that they made a series out of it, just like they've done with Game of Thrones, will be airing today. So I've been getting very excited about this day for various reasons, but you will have to indulge my slightly book nerdy references in titles of slides. They are references to that book and a tribute to it. So as I was starting out with, I will be talking today about tiny genomes, and the coronavirus genome has a tiny genome. It's only 30,000 bases long, which means that if you use amplicons of more than 1,000 bases, you can tile it with just 30. This is quite special because that means that we can amplify the genome of the virus directly from clinical samples, where historically you would need quite a bespoke amplification protocol or bespoke primers. These protocols came together very quickly. First off in the UK by the Arctic consortium by Josh Quick, and they were adapted right here in New Zealand by Nikki Fried and Olin Salander into the Midnight protocol because it's 1,200 bases, so it's a nice reference to that. Allowed us to really quickly amplify these viruses out of those samples and not just look at one part of the genome, but at the genome in its entirety. We coupled those amplicons with nanopore sequencing, and one of the reasons why we pushed for Oxford Nanopore sequencing is one, because it allows for smaller volumes of samples to still be economical, but two, also, and this is the big one, that it allows you to start looking at data as soon as you start sequencing, basically. So some of our reports we issued within 1 hour of starting the sequencing reaction, which meant that in terms of being able to receive samples from the diagnostic labs and turn around reports, we have had turnaround times less than 12 hours, which included the wet lab work. And as James Ferguson eloquently put it, is that you can do the same thing with Nanopore as you can with Illumina. You can just do it in hours rather than days. And they showed that it actually has the same sort of outcomes in terms of the actual bases that you read, because some of the things that people still wonder about is the actual base accuracy good enough from Nanopore, especially when you're dealing with tiny genomes that you can sequence to 300 depth. That definitely is the case. And also more recently, we've been looking at what they call the super high accuracy base calling model from Nanopore. And actually we see that even for some of the larger bacterial genomes, we can get very, very close to Illumina-like quality in terms of actual per base fidelity. So some of you might have heard Gemma's talk the last time round. So I will not spend too much time on this, but for those of you who haven't heard, is why would we want to sequence these genomes of these viruses? Well, they can help us understand an outbreak. It's an additional and independent line of evidence. People, they have selective memories, or sometimes not even the greatest of memories. The virus can't forget its mutations very easily. So it's a different line of evidence than what you get in terms of when you ask people questions. It can help you estimate the number of introductions that you must have had, especially in our first wave, and also going forward when our borders reopen. It gives you an idea of how many unique introductions of the virus you have coming into your country and sort of what the magnitude of that is. How transmissible is it? The fact that we now all know that Delta is a different beast is because we know that it is much more transmissible than sort of the OG COVID version that first originated in China and went to Europe. And that is one of the reasons why this is a lot more hard to control. So it tells you something about how fast it's spreading and if it's changing. And also in terms of where is it spreading, what are the locations, what are the settings that it's spreading in. So it really helps you understand what sort of disease you're dealing with, which wasn't necessarily clear since that this is a disease that we've known about for now almost 2 years, which is not that long in the grand scheme of things. And the other thing going forward is that it will allow us to study if the virus is starting to escape the vaccines. Is it changing in a way that means that the vaccines become less efficient in stopping it? Now, how we do that is through a process called phylogenomics or phylogenetics, where we look at changes in the genome. And the luxury for us in this one is that because the original sequence was obtained in China, we know what the ancestor of this virus looks like. So we can quite eloquently compare it to that and see what the differences are. And then any changes that are present in all sequences must be older than changes that are only present in a couple of sequences. So in this example here, you can see that the blue variant is present in all these genomes. And that is where we then root the tree. So that is where it all started. Whereas the orange variant is only seen once, and that has happening in that purple-red background. So if we would find a new case that has both the orange, the red, and the blue variants, the most likely place where it got of that infection is probably either case E or slightly further up the chain in case D. So that is how we can sort of reconstruct the history of the virus. And it also helps us identify sources and transmission chains. COVID has become the most sequenced organism since Phix, and there's a little Illumina joke in there for you if you appreciate it. But Rob Lanphier is one of the people that has really been pushing the boat out because, as you can see, the GISAID counter that I grabbed yesterday, there's 5.2 million genomes of this particular organisms currently in the databases. And most software that we used quite clearly and quickly broke with those types of numbers. So the software innovation, sort of after the technological challenges of sequencing this thing were settled, there was a massive push for software innovation, and it has, as many times before in the field of genomics, been an important part of the advances that we now have. And the other thing to note there is that you can even view these trees in your browser with over 5 million tips on their ends, which is quite impressive. And Rob Lanphier refers to them as quantum leaps, but I do think it is important to note that this is truly a different scale than what we were used to working on. And due to that, and due to also the investments that have been made globally, we now have all the tools for COVID. If you want to know where a case sits on the global tree, you can know that within minutes. You upload it to one of these servers, and it places it on the tree, and it gives you a nice visual of where that is and what regions those other cases are coming from. There's a real-time view of which versions of the virus are circulating in a certain area or around the globe. So we first had Alpha and Delta. So here on this panel I'm talking about here, you can see that over time on the x-axis, how Alpha was displaced by Delta. It's much more infectious brother, if you will, and how both of them clearly outcompleted the OG versions, the A and the B versions of COVID. And there's all manner of dashboards in terms of mutations in the spike protein, the protein that is used in most of the vaccines, at least the mRNA ones, where you can see how that is changing and what the defining mutations in that are. And those are of interest for vaccine escapes. Again, all of these things are being updated on a daily basis with the thousands of genomes that are generated each day across the globe. So this is also a slightly weird position to find yourself in, where there's these insanely large international groups that are building all these tools basically specifically for the thing that you're trying to do. But as I said, we didn't necessarily knew everything about COVID, even until a year ago. One of the things that you might remember is that we talked a lot about fomites and possible surface contact transmissions. We were all told to disinfect surfaces and to do hand washing and a bit about sneezing and covering our mouth. And as you might hear recently, that has changed. We are now hearing things about ventilation and mask wearing and all those things. So something has happened in between that time that made us think differently about COVID. And this is a bit of a story about what. One article put it quite bluntly, but truly, is that there was a particular scientific screw up that helped COVID kill for a while. And that is the discussion about what is airborne or what constitutes an aerosol. And as you can see here in the visualization made by the BBC based on WHO data, it is considered that something that is less than 5 microns can be airborne. COVID particles, however, are larger than 5 microns. And therefore, for a very long time, people said, no, it's not an airborne disease, but it is. So why did it take so long to accept those facts about COVID? And luckily for us, epidemiology and genomics were still working, even though the physics community might have been having a bit of a debate about what can fly and what cannot. It reminded me a bit about the story about the bumblebee. There were plenty of epidemiological studies that kept pouring in, all of them showing that COVID was primarily spreading indoors, poorly ventilated areas, airborne transmission, long distance transmission, all these things that are hallmarks of an airborne disease were being found again and again. And we like to think that we were part of that evidence base. And some of those findings we found early on, and I will share some of them. And that is because we were in the unique situation where after our first wave of COVID, which you see here on the left-hand side, this is a timeline of all the genomes that we've done for COVID. that are colored by the type of virus, the lineage that it derives from. So certain colors designate a certain family within the virus. Where you can see that in the first wave, there were quite a different number of viruses present by the different colors. But after that, we had a very successful elimination strategy, and we only had things coming in the border. And so that is sort of that background noise that you see going on with the different colors. There you see that light blue peak. That was the first Auckland outbreak that we had, and we successfully eliminated that. So you won't see that sort of same virus come back again. But we were in that unique situation that we had a very good understanding of what was coming into the country. We were sequencing every single case that came to manage isolation, and we were sequencing every single community case. So we had a very good understanding of what was happening. And one of the things that we saw was that on a particular flight, there were a bunch of genomes that were very similar. And typically when people come from different parts of the globes, they tend to have different versions of the virus, just because by that time it had time to mutate. This was sort of March 2021. The thing is that these people, based on their travel data, came from vastly different parts of the globe, almost as far as you could design it if you were designing a scientific experiment here. And there was no clear indication that they had any sort of shared exposure in the transit hub in the Middle East, where they all transited from through New Zealand. So this looked to us as a case of in-flight transmission. So there was some more digging done. And actually, if you look at the disease onset timing, as well as the seating plan on the plane, it seemed to be quite obvious, at least to us, that those two cases in blue, A and B, seeded an infection during the flight, which is not that surprising on such a long haul flight, that something like that might happen. To at least 4 other individuals that were seated in close proximity to them. And again, because we had very rigorous testing in MIQ, we also knew that the other people that were slightly further away on the airplane did not become infected. And also the fact that they were not necessarily sharing rooms or quarantine facilities strengthened the evidence. Now, we were able to get this out in a preprint fairly quickly, which was pretty good timing, since that on October 15th, 2020, there was a study that came out from a US agency that basically concluded that catching COVID on an airplane was nigh on impossible because of the filtration systems they use in the constant air circulation. And the media took that quite to heart, because there was a big holiday coming up in the States and people were making all kinds of plans. We were able to get that preprint out quite shortly afterwards, and it actually on the 19th it was up on our website, and it was in quite good collaboration with the Ministry of Health, which is something that was quite an interesting collaboration to get that out and to get it to the right people. And actually meant that this is now one of the most cited articles that we publish, not necessarily scientifically, but in terms of the news outlets. All the major US news outlets took this up as sort of a cautionary tale to people that were planning to travel, about that it wasn't something that you should just take for granted, that you were not going to get COVID. We had another tricky incursion, again, something similar, where we saw genomes that were a bit too similar based on what we knew about the context that those people could have had, where with the public health unit and the lab that was running the actual quarantine facility, with a very good collaboration, where they went and reviewed CCTV footage. Initially, the news outlets made it out that it was the bin lid. And again, this was still in the time that we were mostly talking about these fomite transmissions that were to touch or surfaces. But it actually transpired after a bit more of a review is that there was a opening of doors within the MIQ facility quite rapidly after each other. And this was the more likely explanation for what has happened. Because what we saw is that cases A and B, again, we love these letters to indicate some sort of order in time, had seeded a infection on the airplane to case C, and they were adjacent in the MIQ facility next to case D. And that was the only contact that that particular case had with any people that traveled on that airplane. So that's why it must have been through that interface. They then subsequently were repatriated within New Zealand because they were infected quite late during their stay in MIQ, and infected additional people on their second flight back up the country. And those people then brought it to their respective households, meaning that we had a couple of community cases that were linked to that MIQ instance. And again, it was sort of those deep understandings of that case D must have been infected by case C, and that there was that very detailed review of the CCTV footage, seeing those doors open and closing in quick succession, that allowed us to make those claims that this must, once again, must have been a case of aerosol transmission, since that there was no direct contact of any way or form between these people. So something's clearly in the air, and some people in Spain did a great visualization. If you ever want to see an example of how to communicate science and the ventilation, they were starting to tell these stories about how rooms and bars and classrooms, how these were the places where COVID was spreading. And this is also one of the things why you now hear a lot of talk about ventilation and mask wearing in terms of when we can back to work under these new traffic light systems. It is going to be a prime suspect in terms of trying to reduce transmissions. But we were doing well. We were eliminating the virus. When there was an outbreak, we were using science to get on top of it quickly, to have minimal disruptions. But something happened. Delta came along, much more transmissive, much more infectious, much higher viral loads in people that carried it. And countries that were doing really well started to buckle under the pressure. And as these things go, it came to New Zealand as well. So we were dealing with these in a different way. And as you are probably all very aware, we are in the middle of the largest outbreak in New Zealand ever, even within the different communities, not just Auckland, but the rest of the country now as well, in terms of numbers of cases, where we have all these stories going out. And if you're interested in a bit of the first write-up that we've done, just a couple of days ago in the Herald, there was an interview between Gemma Geegan and myself about what we found. And I'll share some of that with you here today, but if you want to just read that at your leisure, it is the link is there for you. And just credits to James Hadfield for yet another great visualization of what we're dealing with here. But where we seem to be doing really well, we were repatriating our people from Sydney because there was a large outbreak of Delta ongoing. But something slipped through the MIQ system somewhere. We still don't know quite where. We have a couple of smoking guns in terms of the people in MIQ having the same genomes as the people in the community. That particular genome being present in Sydney. But there's never sort of the missing link was never conclusively found. But there is a balance of probability that something leaked through the border when we were repatriating those people. And we had the first transmission happening within these social networks. And there was one event that was linked to a large gathering of beliefs and events up in Auckland, the Manjari church cluster, as it's referred to often in the media. We were genomically lucky in the sense that there was one particular event right here, if you can I hope you can see my cursor. If not, you will have to focus just on the purple dots. Those are the ones that are epidemiologically associated with that cluster. But the fact that that mutation, that was the one that was seeded at the super spreading event, allowed us to track that quite precisely, because any descendants of that also carried that mutation. And actually, that was a very large part of that first part of the outbreak. There were a lot of people that got infected either because of their family members or because of that event. And the gatherings that were around those events. The good thing is that there was a really strong compliance by that community. They really rallied behind the cause. They adhered to the lockdown measures. And at some point, we were convinced that that part of the outbreak had been extinguished. We weren't seeing cases that had that particular genotype for days on end. While we still saw that sort of that original version of the virus still ticking along in the background. Those were the cases where it was harder to get to, and where you have all have heard in the media that there were other circumstances, meaning that these people might not necessarily be able to stop working because they were actually essential workers, or they didn't have those luxuries that others had. And it is that part of the outbreak that continued simmering. And here I'm focusing on the latter part of the outbreak, the more recent part. That is the part that ended up finding the inequity. It found the housing crisis. It found the crowded housing situations. It found all those other things that we know historically have had an adverse impact on people's livelihoods and well-being. COVID found that, and it spread there. At some one point, a deletion happened, and that's in this part of the tree over here on the right-hand side, after which you see a lot of those orange dots, where that was spreading across Auckland, and we saw it popping up everywhere. It wasn't one neighborhood, it was widespread. And then as restrictions started easing, that is the part of the outbreak that really flared up. And as you can see here, there's a large number of dots here, which means that they're all identical genomes, which means that there's been rapid turnover, rapid infections happening, possibly, again, super spreading events within households and other settings where there's a high degree of social contacts. And the church cluster did have a small resurgence, so we were not correct in assuming that it had been extinguished completely. So even within those communities where there was that really strong adherence, Delta still managed to trickle along in the background, and we still have a couple of cases that are still linked to that, genomically at least, to those events back in August, that are now still causing cases. Some of the ones that you might be of interest in, and where, again, I can sort of show how the genomics helps us understand these transmission chains, is the cases that were detected in Taupo. What you can see here on the left-hand side is the sort of the geographic distribution of those cases. And on the right-hand side, again, the genomes. And what we see is that there's a part of the cluster in Auckland that seeded those events slightly further south in the Hamilton region. And then subsequently, some of those cases were in contact with people that traveled back to Taupo, where we now see cases that are all clearly linked to that part of the outbreak. So you can sort of see the virus starting to skip towns and make its way across the country as people travel and unwittingly take the virus with them. The other one is the one that it ended up in the Wairarapa. It was there was some speculation if that might also be linked to those cases that made their way to Taupo. But actually what we saw genomically is that it's a different part of the cluster, which is more associated with those poorer health outcome populations and housing arrangements. where it's that part of the cluster that spread within Auckland, and then actually someone must have been in contact with someone somewhere, making its way all the way to basically the valley next door for me, the Wairarapa, where we now have some more cases and some more genomes will be coming in today. But these types of things help us understand how the virus is spreading, and also helps us understand how those social networks are connecting. And you might have heard some of those studies that were looking at the interconnectedness of Auckland, and that is something that we are seeing here as well, where there is those between communities social networks that are very strong. So this is the picture of today's tree. You can see that it has spread across the country. We just had a reintroduction into Christchurch. We've heard from cases in maybe in the Palmy region. It's spreading, and it will continue to spread. It's Delta. It is very spready, if you will. And as we head into summer, we all have a part to play to minimize that. So without wanting to sound like a plug, if you have anyone left in your extended family or context that either doesn't care about wearing masks or hasn't engaged with the topic of vaccination as much as they could have, this is an excellent opportunity to talk with them in terms of what they want their Christmas to look like. Do they want that to be in an overfull ICU unit, or does that want to be in a appropriately socially distant outside setting with their family? In terms of some other topics that I just wanted to touch upon is that you might have also heard a bit about the wastewater testing. And that is something that has also been set up in the last year by Joanne Hewitt and a lot of fabulous people in ESR. Where we really wanted to push out the boat to have at least an early warning system to detect incursions. And you've seen in Taranaki that it actually worked. The wastewater was positive before we had confirmed cases. So keep an eye on that. But it will also be a useful tool going forward, since that by doing that wastewater collection, you can actually sample a lot of people in one go. And that also allows us to do the genomic monitoring in a more economical way, since that if you have a mixture of cases, you can actually distinguish the different genomes there. So to sort of prepare ourselves for that, we've been monitoring the wastewater at the jet park initially as a sort of a positive control. But because we were sequencing all the clinical cases that were present there, we could also see how well we were at picking up those different versions of the virus in the genomes of wastewater. So here's an example of one particular lineage that is called B1.170 that came to the Jet Park Hotel in December last year, where the bottom panel here, the more bars that you show there, the more the genotype of that particular lineage has been detected. So if it's fully colored, that plot, that means that you have 100% B1.170 in the wastewater. And what you can see is that there are a couple of positions that are also present in other lineages. So you see that prior to the arrival of that particular virus in the MIQ facility, so those are not sort of unique positions, but they're a nice positive control for the presence of COVID at all. Then there was some wastewater that was negative completely, because shedding through feces and other bodily fluids is not as consistent as it is in your throat and nose. But from the 11th onward, we started picking up signal that was specifically for that particular lineage. And on the 12th, we had a full genome, and on the 13th, still sort of half the genome of that particular lineage being present in the wastewater, which coincides sort of what the predicted shedding pattern for that person, that sort of its peak of shedding is when it was actually picked up. So this was a nice sort of positive control and proof of concept that we can detect these specific versions of a virus in a mixture of genomes, because you can see that there were plenty of other genomes present within that facility at that time as well. It also brought us some really good opportunities for outreach. We were invited to sing Aotearoa to talk about how we were using genomics in real time to inform decision makers. But we're nowhere near as prolific as Mr. Baker and Susie Wiles, but between Gemma, myself, and some of the others in the Phylogenetics team, we did a fair bit of interviews. We were doing things with Puhura STEM Academy and all kinds of other things. And I think the other thing that people shouldn't neglect is the fact that we had our Prime Minister and Director General of Health talking about SNPs and genomes on national television. If there was ever an opportunity for you to engage with the communities that you're working with about genomics, don't miss the opportunity that this has created to open up that conversation about what genomics can do. And we've been working together with a local studio in Dunedin to make a story about what we've done with COVID and genomics that takes a Tau Māori lens to it and tells a story in a way that would be accessible to kids, at least younger aged people, to tell the story of what we've been doing and hopefully get them to engage with the topic. The other thing for the slightly more scientifically savvy, with every publication, or at least the big ones that we do try to make a publicly accessible narrative. Again, using NextStrain, thanks to James Hatfield, where we talk you through the findings that we have. So this is one that we did on the different border incursions. And again, it provides people with an opportunity to interact with the data and to see the story being told by the people that actually did the research on it. And to come back to my statement at the beginning that tiny genomes can make a big difference, when some of those lockdowns did happen because of certain links being found, that helped people stay safe and healthy, because lockdowns are not great for people's health and well-being, but it also helped them keep safe from the virus. But it also protected the economy. And a scenario has been run where they looked at real-time genome sequencing in wastewater. And the fact that that was readily available during some of our important outbreak scenarios is estimated to have saved the New Zealand economy over $2 billion. And that's mostly due to the large effect of what they call voluntary lockdown behaviors that people will adhere to when there's greater levels of uncertainty. Something that we will all get to experience this summer, as there will be a large amount of uncertainty about what things look like. All of this work was funded by the Ministry of Business and Innovation and Employment, the Ministry of Health and ESR. And I just want to acknowledge that all of this was done in an excellent collaboration with the wider science community. This is not just an ESR party. And I'll be happy to take any questions while I leave the acknowledgement slide up.
So thank you. That was an awesome talk. And thank actually for all the speakers in our seminar series this year. This is the last seminar for this year. We're hibernating for summer. We will be back in February. We'll send you around the list of all the awesome people who will be talking in our seminar series next year. But thank you all for coming along. I hope to see as many of you as possible face-to-face one day, but otherwise have a really safe summer. And we'll see you back here as soon as we start up again. So thanks very much. Thank you for an excellent talk. Goodbye. Have a good summer.